put together. Okay, so I'm gonna get this show on the road. Hey, everybody, I'm Terri Conneran. I'm with KRAS Kickers, and I am really happy to see everybody here tonight. We're a good looking group of folks, and you guys have absolutely no idea how incredibly excited I am this evening to have our guest. Okay. Dr. Kevan Shokat is one of the reasons that some of our folks are still actually here. He's a professor and chair of Department of Cellular, and bear with me as I just read this here. Cellular and Molecular Pharmacology at the University of California, San Francisco, professor in the Department of Chemistry at University of California, Berkeley, and an investigator with the Howard Hughes Medical Institute. He's being recognized this coming weekend for the AACR Award for Outstanding Achievement in Chemistry and Cancer Research. And this award is presented for outstanding novel significant chemistry research that's led to important contributions in basic cancer research, which is us, okay, which is KRAS. And he also gave me what I consider my own little Oscar, is that he sent me this, which is like, okay, what I'm going to let him explain to you what this is. And he's smiling here because he got a little KRAS Kickers swag, and I got this. And it says right on here, KRAS is not undruggable, from K Shokat to KRAS Kickers. And when I got this, and it came in the mail, I was by far more excited than when I actually received my Christmas present from my husband. And because I knew I was getting a Christmas present from my husband, I didn't know I was actually going to get a 3D model of what a RAS protein is. And then I come to realize this thing is almost identical to the same size as my lung cancer tumor was when I was diagnosed.
Whoa.
And, yeah, right. And so, and when that hit me, I mean, I got to tell you, I was like totally blown away. And so his dance, so Dr. Shokat's dance card is like not just crazy cool because like he's just this fun dude, but because he's like just incredibly brilliant. And if you look up any of the stuff that's made KRAS druggable, his name is attached to it. And so thank you for joining us tonight.
Thank you, Terri. That was a really, really kind introduction. Thank you. Yeah, thanks for inviting me.
Well, we want to hear what you have to say, and I can literally just pelt you with questions all night long, but I'm just going to let you just kind of like roll with it, and I'm going to let you set the pace, and let's see like where things go, if they're okay.
Sure. You know, I'm thinking maybe I will put up a few slides just so we have a little visual. I really think that you know, showing what the molecules look like maybe helps people see what I'm explaining. So let me do that. And I do want to start with something that maybe people that present to you guys don't start with, but I think it's important. You know, as Terri said, I work at the University of California, San Francisco, but I also am involved with a number of companies that are trying to make drugs to treat KRAS tumors. And so as I tell you the information, I first want to tell you that I have a financial sort of conflict with those companies, but I'll also explain sort of why we work with companies, because I think that is necessary to get the drugs all the way to patients. So those are my disclosures there. And I'm going to jump around in this talk, but I want to start with this. So when we started working on KRAS about 12 years ago, we sort of took we took several unconventional, we made several atypical decisions in cancer drug discovery. And the first was to we wanted to be focused on KRAS because of all the things you know, that this has been a very hard tumor type to treat, and other drugs that we use that block the pathway that KRAS is in don't have as big of an effect if we put the drug right on the protein. So that's what we really needed to do. But the problem was that the protein RAS didn't have an obvious pocket that we could design a drug. Well, you know, we think of it, the protein as the lock, and we think of the drug as the key. And this lock RAS didn't really have a nice place for a key. So people thought, well, there's no key that you could ever find for that. So we went and we looked at a diverse set of tumor types. And these are just sets of tumors that are commonly mutated with the blue dot that have KRAS mutations. And I'm sorry, many of you will know this is a mistake already in my slides. Pancreatic cancer is the one that has a high frequency, colon and lung. And when we look at the change that happens in KRAS from the normal cells to the cancer cells, this one position, the glycine at position 12, gets mutated to 3 different amino acids. So changes in the base pairs of the DNA ultimately give rise to a difference in the amino acids in the protein. And the glycine 12 position can get converted into aspartate or valine or cysteine. And we'll come back to this a lot. But these 3 different amino acids, they roughly create the same kinds of tumors, but they have different chemical features on them. And the most common is aspartate. When we go to colon cancer, the order is still the same, but aspartate decreases, the slice of the pie decreases from like 3/5 down to 1/2, and cysteine starts to increase. And when we look at the lung cancer frequency, the cysteine grows in, aspartate decreases in valine. So now why am I telling you so much about the names of these amino acids? And that is because each amino acid has a different chemical property, a different kind of reaction that we can use to sort of sense its presence. And if I take then all the tumor types and sum up all the frequencies across the different KRAS mutant tumors, aspartate is the greatest, then valine, then cysteine.
Can I ask a quick question?
Oh yeah, please, I meant to say, interrupt
Okay,
away.
all right. Well, don't tempt me. I'm good at it. Okay, but those, the cysteine, the aspartate, those are where we're getting our letters, our G's, our G's and C's and D's, right?
Exactly, yeah. So this would be D is for aspartate, V is for valine, and C is for cysteine. Exactly. Yeah. Yeah, when there's the 3-letter code for the 20 amino acids, and then there's the single letter code. Yeah, exactly. And so since the discovery of KRAS and all the other RAS mutants, people have noticed that aspartate is the dominant one. And so people were trying to make drugs against the aspartate, but that has some challenges, mostly a lot of it because the pocket was never visible in RAS, so we don't have a key for it. And if we want to take a sort of unconventional approach and not just sort of use what we call reversible binding, so a drug that can bind and it can unbind, bind and unbind, that's reversible binding. Another kind of drug, sort of the way aspirin works, for example, is when it binds, it stays stuck. It makes an irreversible bond, so it cannot come off. We thought, let's try to make one of those kinds of drugs for KRAS. And the best one to start with was the cysteine, or the G12C, because that amino acid among the 20 natural amino acids is the rarest and it's the most reactive. So it gave us the biggest chance to sort of make a drug that would find the needle in the haystack. And the great part about a strategy like that is that if we made a drug that relies on the cysteine to bind to KRAS, and it binds because of that irreversible bond, it will not bind to anything that doesn't have the cysteine. So all the other RAS, the normal RAS, the glycine 12 containing RAS in patients' bodies, will not be inhibited. So it will be targeted just to the cancer cell, which is a huge difference between all the other targeted therapies that we normally talk about. So they're called targeted because they're kind of selective for one target, but they're often present in the tumor cells and the normal cells. Our strategy is a drug that only works in the tumor cells. It's almost like a little tiny chemical immune system, because the immune system is very good at finding out the difference between one amino acid and another. This drug is also like that. That's what also makes it very special. So once we made that decision, and a lot of people criticized us, they said, oh, you're just going to go after the cysteine, but what about all the other RAS forms? And what I'll tell you is that you need to walk before you run. You need to have some success and learn a lot. And then you take those learnings and you build upon them. And as long as the foundation is solid and that things build, then you will reach for the next amino acids. And so I'll tell you maybe about how that is going as well, which is going very well right now. Still not in the clinic, but very close. So that I'm very excited about. So we first want to look at our target. And the way we look at the target is to solve a crystal structure of the protein. And so before we started, we said, let's look at the lock. Let's find out what the lock looks like. So we solved the crystal structure of RAS, and here it is with its nucleotide bound. And it has these two loops on it that are called switches because they switch between the GTP state and the GDP state. So I haven't told you about nucleotides, but nucleotides are the sort of energy and signaling little molecules in our cells. And RAS binds to two different nucleotides. One is GDP, or guanosine diphosphate. Here's one phosphate, here's the second phosphate. And it also binds to GTP, which has 1, 2, and then a third phosphate here. So we kind of knew all this before my lab started working on it. All these structures were known, and that was very, very informative. But when you look at this protein, what you don't see is a cave. You don't see a nice deep cave, which would be the lock that we can make a key for. And so we said, that's okay. Some of this protein is mobile and it's moving around a lot. And if we take a snapshot, it's just like if you take a snapshot of somebody running, you don't see their legs and what they really do. You just see one moment and you don't really know that the legs are working like that. So we just said, okay, hold off, let's keep working. And then we did some chemistry, and that's where I skipped around because I think probably what we basically took is we have boxes of keys around the lab, and we put them into the protein, and we looked for one that would bind a little bit, just a starting point. And we found that. And then we wanted to now look. And so now this is the protein structure of the drug. The first drug we found bound to the protein. And here's the drug. It starts, and it is a covalent bond between the cysteine. So this is the 12 position of KRAS. In normal cells, it has a glycine. And in the cancer cells, it has a cysteine. And then we have a chemical bond here between a protein and our drug. And you see the drug now has different atoms and different groups on it that sort of shape the key. And it fits into this pocket that nobody had seen before. So this was the breakthrough. I always go back to this slide because this one is where we first saw the protein fold around the key. And it's sort of like a lock that nobody could ever figure out how to open until you have the right key. And we couldn't even see the lock had a place because everything molded around the key. And that basically then led us to race now to the clinic. And that is what we had to do. Now we have to do a lot of chemistry around this drug and make it fit. You see how in here, there's some, I know it's not 3-dimensional but hopefully the shading gives you an idea of little pockets.
I
And so
I can hold up my little thing here too.
Yeah. I made it the same colors as yours, Terri. Yeah.
Right. Okay. So I don't know. I don't know if I'm on screen or not. I can't see.
Yeah, you are, or at least I can see
Am
it.
I holding it up here? Okay. Like,
Yeah.
right. All right. So
Yeah.
let me ask you questions. If you don't mind, like doing this, like we can tag team a little bit. Is that okay?
Oh, of course.
Okay. So like you showed us like that's 12. How do you know that's 12? And if that's 12, where's like say 13 or 61 in that slide? And then how do I see like what this looks like? Can you guys see this? Almost looks like a little bit of a brain. I mean, it's very like three-dimensional. It's very textured. Like, so there was like not like a place, it's just like, you just pop it right on, right?
Yeah. So what you're looking at there is the surface representation of the protein. So inside of that are all the amino acids at the 13 position, the 61 position, that are packed together. They're so well packed that there are very few little water molecules that can even get into the middle of that. So it's a very solid piece of matter that's all folded on each other. And then after it's folded, we put a sheet, if you will, over the surface, and that's how we know the contours.
So, okay, so give me a little bit of perspective, like how big is this in like real size, like compared to like a hair or something?
Yeah, so that is about 800 angstroms wide. So that is like, probably on the order of a 10th or 100th of a hair.
Okay,
so you can't see it. You can start to see it under a microscope if you make a crystal of it.
Mm-hmm.
But it's very, very, it's just, each of these is an atom. So it's not subatomic, but it is, it is smaller than any, human hair. Yeah.
Okay. And then, all right. And so if that's 12 on your slide, right? Okay.
Yeah.
So like, there's got to be some logical numbering system.
That's right.
Through that, right? Because I'm picturing like, when we played like Battleship or something, right?
Yeah,
so,
exactly.
Right? Is it kind of like that?
Yep. I'm trying to see. Okay, you see my mouse?
I can. Yeah.
Okay, so that's 12. And then you see this string a little bit like a spaghetti noodle?
Yeah.
Okay, the next position would be 13 and 14 and 15. I didn't put a little marker there, but that's where they would be. This is sort of in between a cartoon, and I highlighted just the atoms of the important pieces.
I appreciate your putting up with my questions, just kind of interrupt your whole flow here, but I mean, it's been a long time. I mean, I had other things in my mind when I was studying biology, which was back in high school. So I know that tells you too much about me, but that's kind of like where we're at. And this is kind of like changed. So when you find this and you're describing, you like lay like almost like a sheet of something over it to kind of like see what the texture is. I'm picturing like a skinnet, like you put on top of a car or something. Is that kind of like what you're forming it with?
Exactly. Yeah, this is just a model that it takes the crystal structure that we solve and put it into a way that we can visualize it. Yeah, and so that just lets us sort of, I could pull up another view, and I can do that later, where we can zoom into anywhere of the protein and then highlight all the atoms and all the waters and everything. But those are the kinds of things that help us then make the next chemical to inhibit better.
Okay, cool. Thank you.
Sure. Any other questions I can? Oh, I think Ellen, you're on mute.
Yeah.
I think Ellen was on mute, but I saw her lips move.
Yeah, Ellen, you were talking, but we couldn't hear you. Maybe she's on her phone.
Oh, okay.
Maybe she's talking to somebody more important than us, I guess. I don't know.
Okay, when she gets that. Okay, so so this is the drug, and this is, I think, an interesting slide because it tells you sort of how long it can take to once we discover the prototype, the first drug, how long it takes us to get a molecule that's optimized for clinical trials. So this is the one that we discovered and reported in 2013, which was the first picture of the pocket. And so then a company we started with that and then optimized the molecule. And you can see that it's changed, right? The top one and the bottom one, they don't look identical, but there are pieces of them that look very similar to each other. This is where the cysteine is going to add, the same group is there. This is sort of fitting into the pocket, and that looks the same. And that took about 18 months. Then it took about 3 years to go from this one to this one. And now you could tell it looks quite different. And a lot of that has to do with just trial and error, and then solving more structures, and then doing more designs. And this is probably the work of 4.5 years, 5 years of people, and I would say about 70 people.
Wow.
So it's a significant, when you see the answer, it doesn't look like it's that hard. But to go from here to here, it took 800 molecules. To go from here to here, it took another 800 molecules. And every time you get the molecule bigger, you have more chances of breaking something than you have of making it better. So there's a lot that goes into that.
So, is all of this happening like, right? Okay. So like, walk me through like where this like happens. Cause I mean, you know, as a patient, it's like, we don't know. And then all of a sudden you get diagnosed and you're like, okay, well, why is it taking research so doggone long to get stuff out here? Right. Okay.
Such a good
and this was like 5 years. Okay. And it took,
yes.
and it looks like, I'm sorry, squiggles on a piece of paper. And, you know, and you're like, oh, it's 800 atoms, which is almost as meaningless as squiggles on paper to me. Right. And so it's like, this is hard stuff. Like you've got some sort of fancy machine that you're doing this with, and this is in like little tiny, like what, pipettes and stuff?
Yeah. We are, each of these were getting made in, you know, we would make like a gram of each test molecule and then test it. So, and then if it worked, they would keep using it. So you're doing a lot of chemical synthesis. And so this went on in my lab with 2 people in the lab, a graduate student and a postdoc. That led 5 years to this molecule. Then the company started working, and that was about 40 people. And to be honest, when we tried to improve this molecule in my lab, we couldn't do it. We kept breaking it. And it was getting very worrisome. But then when the company team got working on it, they kind of looked at it from a slightly different angle, and they had more chemists so they can make more molecules, and that trial and error gets more successful. And so that was done in San Diego at a company called Araxes. And it was in partnership with a pharmaceutical company called Janssen. So they're based all over the world, J&J. And we had a lot of the chemicals synthesized just outside of Shanghai in China. And partly the reason for that is that we're in different time zones. So they're working during our night. We look at their data during our day, and then we tell them, make this molecule now. And then they send it to us, we test it. So we get to work on a 24-hour cycle because of that advantage.
That's kind of cool. I didn't know that that's how that happens.
Yes, for our companies, we've done it that way, and it was great. It started as we chose to work in China because it was less expensive. But then when we saw that advantage of the 24-hour cycle, we were like, we're not going back. Yeah.
Because I know in general, like scientifically, there's like, we look at like data that comes up from China, but it's not always invalid. It may not be like, so there's value in this.
Right. And what's great about doing this part of the work, you know, outside of the company, is that when somebody sends you a molecule, within, you know, a few hours, you can double check that it's exactly the molecule you expect. So the quality control is extremely high, and they are fantastic people that we work with there.
Awesome.
And then, you know, to go from here to this I don't think I can explain it, but it needed to be rigidified. And that rigidification is important. So, if a drug is moving around, the key is moving and changing, and the lock is changing, it's hard to make them hit at the right time and be, snapped together. So what we did was we started to rigidify this key so that it was really perfect for that one moment the lock was available. And that's what was the big breakthrough. This molecule now is the predecessor of all of the other KRAS drugs. And you can see the one from Amgen that got approved, sotorasib, you can see it has that same middle kind of orientation. And you can see the one from Mirati, it also has that middle orientation. And they also have the same electrophile, or where cysteine adds, as our original molecule. And then they have these features, which are also similar in all. So it's just, when you start getting closer and closer to the real target, a lot of the molecules start looking very similar because there's just the way things put together. And so, yeah, once, this pattern and this structure was known, then a lot of people, then hundreds more at Amgen, hundreds more at Mirati, were able to sort of even further optimize that. And so this molecule was the first one to work really, really well inside cell culture cells. This one was the one to first work really well in animal models. And then these were the ones that are now working in the clinics. So it just kind of, each step though is a very big hurdle to get through.
So each generation, okay, is it's kind of like, I don't know, like an iPad, right? You get version 1, you get version 2, and so this would be kind of like your original Apple. I don't know what came first, like the Apple phone before like the iPad started coming out, right? And now we're kind of like working our way into like version 12, and
exactly.
Now they got what names of candy is like the types of right, but yeah. Okay, and so as they keep growing and getting better, that's what's going to keep happening, is that the next generation will probably still have a similar basis.
Yes, exactly.
And then it'll target it, because it's like you said, the rigidity. And so I'm assuming it's kind of, I'm picturing almost like a sci-fi movie where it's kind of like fluid and it keeps moving and you got to just catch it just right.
Exactly. That's exactly it. That's the picture we have in our heads. Yeah.
That's well, you did a great job of explaining it then.
Okay, good.
So, yeah.
So now everybody is, and the data from the first clinical trial with sotorasib shows patients that have G12C tumors, and this is their waterfall plot of all the patients' tumors, and these are the ones that respond the best. The tumors shrink the best. And then there's a whole range of activities, and stable, these stand for partial response is PR, SD is stable disease, and then PD is progressive disease. So we want to see something we didn't see in this trial, which was a complete response, a CR. We want to see the tumor completely disappear. But for being the first drug and showing, no question about it, clinical activity, this is a really big accomplishment. And this is what people looked at to say, okay, how do we make a better G12C drug? And how do we make drugs against G12D and G12V and all the other mutants? And that's where we are right now. This is the exciting moment where people are now taking this structure and the other one, everything we know from all the structures that people have discovered, and tweaking them to be more specific for the other mutants. And so we're doing that. Amgen's do, a lot of companies are doing that.
Yeah, it's definitely a team effort. So, okay, so just for fun, at what point did you go, I got something here? I mean, and you're like, like pop the champagne, I got something going on here.
Oh yeah, this, that structure, that came a little before Christmas in 2011. And I opened up the structure and just was blown away and brought my family over to look at it, and I said, this is it. This is the one. There were still many hurdles that we knew about and many hurdles we didn't know about. But once we found this pocket, this is the kind of structure and the picture that converted KRAS from being undruggable to being druggable, because it converted the challenge to a chemistry challenge. And that's something that is like hard to express, but biology is biology. If the tumor is caused by a KRAS mutation, that's what you have to solve. But if you have a chemical that's not quite perfect, you can make alternative chemicals to make them better. And you can, and chemical space is almost limitless because of how you make bonds and how shapes change. And that's what's remarkable about medicinal chemistry. So this is the one I knew that we're ready to turn the corner. That was great. And
did your family recognize how cool this was, or they're like, oh yeah,
yes,
where's dinner?
No, they did. I think they did. They did. I'm sure they didn't quite. I probably told them that before too, but.
Okay. All right. Yeah. And then I know it's always going to come down to like, all right, great. So that's where we are. And then, now we're still going to have acquired resistance. We still have, G12Ds and 13s and, and 61s and so on. Right. And then it's in different parts of the body and things seem to be different. But but we've got the basis now, right? So how do you take that and go to the next step?
Right. What we do is we try to find the target that is got the most number of patients and that we have a good chemical idea for how to solve. And it's the intersection of those two things. So if it's chemically solvable, then we will go after it. And we would put resources on the more frequent tumors like KRAS G12D, for example. But sometimes it's hard to make the leap to the target you really want. You need to keep stepping through. So what we've done is we've done the same thing. I'll show you this. Go back to this one slide I showed before. So these are the main mutants we're concerned with. The cysteine we know we have drugs for, but I think there'll be better ones soon also. Cysteine and serine are very similar amino acids. And so we made a serine, a KRAS G12S drug recently, and that's in getting published soon. We also just in the lab in the last few months made an aspartate inhibitor, and that is looking very, very promising. And these are both covalent bonds, and these were a chemical challenge we didn't even think we could solve. But once we took what we learned on cysteine and applied it to serine, which is similar, and aspartate, which is different but related, we got those nailed. And we've also got arginine ones. And so other people, and I can tell you how people are doing that, we're looking for valine and alanine. But for the covalent ones, the irreversible ones, we have cysteine, serine, arginine, aspartate. So it's just, I didn't think that would be possible for another 6-7 years. But the amount of work going on in the world right now on drugging KRAS, it just gives you so many little pieces to mix and match and combine and then get properties that we need to go after these harder amino acid mutants.
Okay, I'm going to ask like a silly question. All right, it's not, maybe it's not silly, but it seems perfectly logical from my non-biology that I was much more curious about the guy that was sitting in front of me in my biology class than I really was what was going on in the whole class. Okay, so when we're looking at these things and we're starting to identify and like the different names and stuff. Okay, explain to me how this isn't necessarily something like I can eat or not eat, right? That can affect this. What am I trying to say? If they're like, okay, sugar causes cancer to grow. Okay, well, if I've got cancer in a petri dish and I throw sugar on top of it, it's going to grow. That's true
Yeah.
for for yeast too. Okay.
Yeah. Right.
But so how does something like that going to affect like our biology? I mean,
Something like this drug, you mean?
yeah, like something like that, or something that's
Yeah, I think what you're getting at is there's a lot of ways maybe to kill a cancer cell, like starve it of sugar, right?
okay.
But that affects all cells, normal cells, brain cells. We rely on glucose and sugar to sort of think, and it's very important to have drugs that work only in the tumor cell. So that's why what's so special about this kind of targeted drug is that it lets the normal cells do all the important functions that they do. And it takes the tumor cells and deprives them of the key thing that they are relying on to grow, which is KRAS. Now, KRAS isn't consuming sugar itself, but it's telling the cell to sort of grow fast. And the cell knows if I got that growth signal, then I'm going to need sugar. So it's a program rather than the machine. You could think of it as software versus the hardware.
Okay. And so is there like, so it doesn't really impact it then. Like if I give up my dark chocolate, if I stop eating chocolate chip cookies, like that's not going to really impact the tumor then at all.
It's everybody. There were a number of Nobel Prizes given for understanding how metabolism is different in cancer cells and normal cells. But to this day, we still don't have a therapy that relies on blocking metabolism to cure cancer, because metabolism, there's been so long of evolution to sort of respond to missing one nutrient, then we just switch our machinery and we use another nutrient. So it's very hard to make the cancer cell die based on lack of energy. But that's that's my opinion. I just give you the fact that there is no drug that does it. It doesn't mean we shouldn't study it, and doesn't mean we won't find it. It doesn't mean it's not a great idea, but we still haven't learned enough.
Okay. And I know I'm asking what seems like a simple question, but I'm sure there's like so many different levels of complexity that are way beyond.
Oh yeah, but you're showing us, you're using your intuition to follow your curiosity, which that's the key to all science. So you're
Well, and I mean, it just kind of like makes sense, but at the same time, if it was that simple as me just giving up chocolate chip cookies, then how come people that eat healthy don't have
right.
it, still die from cancer, right?
Right, exactly.
There's that simplicity, as well.
That, yeah, that's good.
And
I see, Terri, a lot of questions in the
yeah,
chat,
sure.
but
So
I'm
let me
not
just
keeping
jump
an
in
eye
there.
on them. I don't know.
No, that's okay. Let me just jump back at them. Okay. All right. Question. What can patients do to help accelerate research so it doesn't take 5 years?
Wow, that's a great question. It's I would say, I never thought about that actually. I mean, we think about it at the company and we look for problems where more money can solve to go faster. And you can usually find money to do that. But the bigger problem is all of the experiments we haven't started to get to the starting point of the 5 years. So I personally think that we need the discoveries that get us into that 5-year window. If we had more of those 5-year activities going on, we would have so many drugs. But a lot of times research isn't funded that would lead to the beginning, little seed of a drug. And so we can get things from 5 years to probably 3 years, but it might be more efficient to fund more discoveries at the early science stage, because then that gives us more possibilities, if that makes sense.
And just kind of in relation to that, the KRAS Kickers has a partnership. We're funding research, and so we're And we will, as organizationally, be able to participate in choosing where that funding goes for that grant with Lung Cancer Research Foundation. And so, this is something that we're doing as an initiative. Anything that we do, you guys are free. We don't charge people, but if you want to contribute back to it, that's the way to do it. And if you want to learn about the science, hey, and we need people to understand it from a patient point of view, as far as like realistically, and be able to speak to doing the grants and stuff. Okay, I'm going to move on to the next question.
Now I thought of one other answer to that point. One thing that companies do is that when they're trying to make the drug, they might try, what happens in drug development is we often get a drug that works on the target and like in that crystal structure and works in cells, but to get it to a patient, they always want it to be an oral drug, which is obviously the best. But sometimes making a drug that can be injected IV can be 2 years ahead of the other oral drug. So I think if it was me, I would take the IV drug if it would come 2 years faster. And yet companies often won't make that decision because they think the market won't reward them. And so whenever I advise companies, I make that statement very strong. But that's kind of
well, if I have a 2-year life expectancy, and I'm rather have the drug today than have to wait 2 years and not be alive for it. So just putting that out there. And I know that
exactly what I say.
I see a lot of heads nodding as well. And I know at one point we had a question about how come sotorasib is not like, you can't get it by an IV. And, in chemo you can. So I don't really know the answer to that. One of the other questions that we had run into when we did a thing about research is they were saying that getting like really good tumor samples from patients is an issue. Is that, how do we fix that? I mean, you'd have all my tumors. I mean, what do I need to do to give them to you?
Yeah, no, I think there's a lot of work in that area, and it's gone much better. I think when people say they can't get tumor samples now, it's probably the way it was 5 and 10 years ago, but now it's much better. It could be a case that the major universities have those systems and the smaller places don't. So, but I think if you promoted that, it would be very, very valuable still, but it's not the main roadblock, I would say.
Okay, well, if you have a roadblock or other people that because you know the people that I don't know, right, by far, right? If they have roadblocks, I mean, we have people that are like, what do we need to do? Whether it's advocating for it or we need to sign something when they're taking it out of our bodies, because I mean, I don't know anybody in here that doesn't like, no, just take it out and just do something with it. Okay, I mean, it's not going to do anything.
Yeah, no, that's a really valuable perspective that you all bring that I think could change the sort of perception. Yeah,
and so okay, and then all right, next Lori says, Dr. Shokat, I'm on my 10th month of sotorasib. Today my oncologist told me I am NED, no evidence of disease. Thank you so much for being part of extending my life.
Oh my goodness.
And I want you to know that when I know Lori, and when I talked to Lori before I should make her come on. Otherwise, I'm going to tell her story. And her doctor didn't do the biomarker testing. And so she ended up getting metastasized further. And when I talked to Lori before she was on SOTO and before she knew she was KRAS, I didn't think she was going to see the end of the month. I mean, she was so sick. And today, I mean, I just talked to her earlier today. She got an NED, which is as good as it gets, y'all. I mean, they don't say cured. I mean,
wow.
So this is because of what you're doing. And so when I'm like, no, you don't understand. I get so excited because what you're doing to change like molecules and atoms in this teeny tiny little thing that are like, there's a billion of them for our hair. Okay. that's giving us life. And I can tell you how many stories like this. And so you don't understand how powerful this is. So you guys need to keep doing what you're doing, and your research needs to keep going. And whatever that takes, okay, let us know in the advocacy community, because we can't be here. Okay, it really is the KRAS knowledge and your research. Okay, our advocacy is going to give us survivorship, so we got to do that. Okay, so all right, so question: if sotorasib doesn't work, can one try adagrasib? We're not going to say from a patient point of view, we're going to say from a chemical point of view. How's that?
Yeah, there does seem to be, I don't know of any cases where somebody started on sotorasib and went to adagrasib and had the response. But when we look at the data from the colon cancer patients that have the G12C mutation, the response to adagrasib seems better. So I think that is something your physician would consider, because they are different. And if you'll permit me, I can just show one little reason why, and I won't be able to, I don't have all the slides to show you this, but you remember, oops, I'm sorry, I didn't share right. So we looked at this. These are the two drugs, sotorasib and adagrasib. And I kind of pointed out what was similar between them. So I gave you the impression that they're going to act the same, they're going to work the same. But there was a very big surprise over here. This little part on adagrasib is missing here, and it's missing here. So it's one of the unique features of this drug. And it turns out that this little piece makes a little extra favorable interaction or stickiness in KRAS. And that made this drug a little bit different and a little bit more potent in certain ways. And that's why I think is one potential reason why adagrasib in colon cancer is working a little differently. But it's, it's just tells you each little difference can make, can be important. And we don't know that until we study it a lot. So that was a long answer, but I think I see things in the two molecules that are different. And when there's a difference, then it could be a biological difference. Yeah.
Okay, all right. See, I've read about Mirati's. I think it was the G12D drug is that they're asking about. Am I correct in saying this is not going to trial yet? I do not know that the G12D has not gone to trial yet for Mirati. Is that correct?
That's right. It has not, as far as I know, too. Yeah. Okay. And there's that G12D drug, which is reversible. There's one from a company I'm involved in called Revolution Medicines, which is called, it's a different kind of drug, but it also works on G12D and it's covalent. So that one is going to be interesting. And I think there's a lot more people are just getting ready to release the data on. I was going to talk about mine next week at the ACR meeting, but I'm not going to be able to. We're just still finalizing it. So, but okay. It's moving well, I think.
Okay. Well, and well, maybe we can circle back, even if it's just casually, because you'll be there as well. We can talk about it.
Yeah.
That'd be cool. I actually get to meet you in person, and I am like really kind of like fangirling a little bit. Okay, so all right, this looks a little bit heavier. Do the G12C various inhibitors that are being developed have similar affinities to the KRAS pocket? Let's see.
They do, except that one on the Mirati molecule has a little bit of an extra piece that makes it a little bit higher affinity. And
when you say affinity, please.
Stickiness. Yeah, that's that. Yeah. Thank you. Sorry about that.
No, that's okay. It's my job to be dumb. So it's all good. Okay. Is the drug resistance?
Oh yeah, the drug resistance of the KRAS G12C inhibitors transient. No, unfortunately, when the resistance comes, it usually then is around, and then we need a different drug to get rid of it. And we think that another drug, the RevMed Revolution Medicines drug, it's called 6291. And that one is likely to be different. And that's been published in a cancer discovery paper. And probably more papers are on the way for that. So that's what I mean. That could be another quantum jump in the ability to treat G12C tumors.
Okay,
but we won't know till we're in the clinic.
Let me carry this down just a little bit. Okay, so you're talking about if I'm on a G12C inhibitor, whether it's adagrasib or sotorasib, and then my body starts getting a resistance to it and the medicine stops working. That's what we're talking about when we're talking about that drug resistance, right? And so compared to, and as it changes, it molecularly, as it changes, that's what we're talking about, if the next level of the next version of drugs that we'd be needing, right?
That's right.
Am I tracking? If I'm wrong, tell me. Okay.
No, that's right. The drug, the protein would change again, and now the drug would not work as well. But if we give it a different drug that works in a different way, we can still get a handle on it.
Right. If my Tylenol's not working, I take Motrin, right? That's
right. Yeah.
Like I said, I'm sorry. I got to put it in terms I get. All right.
We even have Terry. I see at the bottom of the screen, one of the participants, Justin Meyerwitz, is a former graduate student of mine, but now he is one of the physicians at Revolution Medicines, responsible for getting those drugs into the clinic. So good, you joined, Justin.
Hey, Justin.
We're so excited for what's ahead.
So are we, Justin? Like, you have no idea. Yeah, you have no idea.
And it's just been amazing to see this whole evolution since since I was a graduate student starting in 2010, I believe. And I remember when John and Ulf, the postdoc and the graduate student, had this incredible discovery. And John is a very enthusiastic person all the time, and just his level of enthusiasm went from his usual 10 out of 10 to like a 30 out of 10.
That sounds amazing. That sounds amazing. Well, I'm really glad that you joined with us tonight, Justin. And, and just so y'all know, I'm talking Revolution Med because I do want to know what's going on. And I do try and talk to stay in touch with the drug companies so that we know what's happening here. So we can bring the information. I'm not going down the next list, but how does that work in relation to like a pathway drug when you're looking at this molecularly? I mean, when you go with the inhibitors and stuff, how does that fall into this?
A great question. These proteins are in a pathway that send information and branch information and check sort of other pathways. So, the cell is, an incredible computer, and your question is, how does it work when we put a little sort of block in one point of the computer? And the real answer is we don't know until we put the drug in there and see how it responds. It's a really, it's something that is so complicated, it's hard to model it and predict what will happen. It's so much more to get what we call ground truth, is to put the drug on there and see what it does. So when we made this drug and we were using it and we were just telling people we were going to put this in the clinic, they said, well, maybe you'll put the drug in and it won't work. Levi Garraway, who's the chief medical officer of Roche, he was the chief medical officer of Eli Lilly. And when this sotorasib was in the beginning of the trials, he asked me on a panel, what will you do if it doesn't work? And I said, well, I'll go right back and make a better one, but it's going to work. So, even at that level, we don't know. So it's a very, very empirical thing still.
I like the kick KRAS attitude there, right? I mean, it's like, well, if it doesn't work, we're just going to go get it back another one. But I know this is going to work. I like that. Okay, that's some serious swag. Okay, let's see. Do the G12C various inhibitors are being developed? Oh, I already answered that one. How the synergy of combination treatment is explained compared to monotherapy? Oh, that's a good question.
Yeah, that's really great. I mean, the best kinds of combinations are ones that cause synergy, which means that, instead of 1 + 1 = 2, we want 1 + 1 = 5. And for that to emerge, we have to sort of know what the first drug does, and the cell tries to respond and prop itself up with something, and then we hit that other drug pathway that's being propped up, and then the whole thing collapses. So cancer cells are not as resilient in a way as normal cells to differing conditions, but we just need to know which way to push them. And then when they get to the edge, kick them over the edge with that second drug. So predicting drug synergy is even harder than monotherapy because, as you could imagine, just the dimensionality gets very huge. But that is what I think, those trials going on now, I think, are going to be very exciting when we combine the G12C drug with other drugs.
I'm really excited about it. I mean, if we're looking at personalized medicine, we're complex beings, okay? If it was a simple thing, I mean, leeches would have solved it thousands of years ago, right? Yeah, okay. All right, yeah, and this I'm going to kind of change this question a little bit. But okay, so how is it like in a lab that you're going to be able to predict, okay, that this looks like it may work better in colorectal versus lung versus an appendix type of a cancer? I mean, is there a way? I mean, is this like you're giving mice like how do you say that?
No.
Appendix cancer? I mean, how does this work?
That is a really great question. And I think that is one of the bigger mysteries in cancer drug development. Why a targeted therapy that targets the same oncogene in two different tissues. In one tissue, we get a great response. In the other one, we get middling or not a good response. And there's a lot of hand waving, and maybe those things are right about why that's different. But I would say at a fundamental level, we still don't understand that, which is unfortunate.
Okay, and so that's part of when we're seeing it. So I'm kind of speculating here, right? So basically,
Yeah.
when we're taking this and you're seeing that it's working in a lab, right, chemically, and it looks like we've got something here, and then we go to like the first round of clinical trials, and we're trying them in patients. That's why we're trying them, and because it may be working in one type of cancer, but not necessarily in another type, right? And then
Exactly.
then you start looking at like some of the co-mutations and whether it's TP53 or tumor mutational burden or these other factors we start looking at. That's when that starts kind of like coming out and filtering down. Is it right?
Exactly. Yes, exactly.
Okay. And I mean, I'm just trying to speculate here based on what it is that you're saying, because I'm just curious by nature. Okay. Let's see. Can we see improvements? Can we see any improvements by 2 months? What is the minimal time for KRAS G12C inhibitor and how do you say the epithelial cancer compared to CRC? I think that's not really sounding like you question. Am I wrong?
I wouldn't be able to give you a good answer. I'm sorry.
Yeah.
I see some people from Amgen on, Lisa and Maria. Maybe they might be able to give a better informed opinion.
Yeah. Okay. Dr. Shokat, I have a KRAS mutation, but my best friend has the same mutation, but NRAS. Okay. We didn't even touch on the NRAS and HRAS stuff. We're both a RAS mutant. Can you explain the difference between KRAS and NRAS? And I'm going to throw HRAS at you too.
Good. Yeah. So when the person said you both have RAS mutations, those are RAS family, and the RAS family has K, H, and N in it. And they're basically, it has to do with how the RAS isoforms are sort of moving around the cell. So KRAS is sort of localized always at the membrane, and N and H, they go to the membrane and they come back. And so part of why one tumor selects a KRAS mutation, one an NRAS mutation, might be something to do with that, although I would say we really can't explain all of that, why one person would get a KRAS mutation and one would get an NRAS mutation. KRAS is the most frequent, then NRAS, and then HRAS. So you can kind of look at all the data, but I still think the fundamental questions about that are still unanswered. Why one family member versus another.
And part of that is too, like, there's the RAS initiative, which is going on. Can you,
Yeah.
would you touch on that a little bit?
Yeah, the RAS initiative is the brainchild of Harold Varmus, who won the Nobel Prize for discovering the source of viral oncogenes as being human proteins. And so way back, they discovered how sort of viruses cause cancer and that the source of those viral genes was our own genome. And one of the genes that were discovered in that period was KRAS. And Harold realized that, we thought we knew a lot about these proteins and genes, and we've solved crystal structures, we know the sequences, we know all these things, but he thought people were getting sort of defeated by the lack of progress on making a drug, and that the reason for the lack of progress is something we just didn't understand. We thought we answered questions, but maybe we didn't fully answer them. So he started with Frank McCormick, an institute, recruited one of my colleagues here to go to Frederick, Maryland, and start a whole building of people that only focus on RAS. And they would do all the experiments that had not been done before and do them to precise standards side by side. So everything is all benchmarked, and go after the questions like this: why H, why K, why N? And really provide a lot of sort of energy to that field. And it has worked. Now we have a drug. So it's a really, you don't hear scientists say we don't understand something well enough, even though it's something that's in all the textbooks. So it took a Nobel Prize winner to go back and sort of make that statement and defend it. Yeah.
Yeah. And so when I regularly am telling people, no, they're working on this. They really are working on this. Okay.
Yeah.
And this is what's in progress is coming. Okay.
And I should have said that, a lot of times when we've got discoveries in the 60s and 70s and 80s, they were discoveries and they were fantastic, but we didn't have the tools we have in the 2010s and 2020s. And so now, revisiting some of those things with new tools, boy, it just blows. You get so much more resolution. You get so much deeper understanding. Yeah.
It's kind of comparing like the old Pocket 110 to like,
Right.
how what is it, 6K, 8K, whatever, right? I don't even know. All I
Good
know is
comparison.
the better the resolution gets, the older I look. I'm not sure what the correlation is on that. Maybe we'll do a project. Okay. And so they're doing again, the question comes up about the G12D, the G12V, and so on, and the 13s and the 61s and whatever. And it's all, I'm going to let you just kind of like really take that and just give me a great answer about it.
Now that we know the pocket and we know, 10,000 molecules that fit in that pocket, now we don't need the sort of cheating of having the cysteine there, which was our big, sort of help to get the first molecule in there. Imagine, mountain climbing. First time somebody goes up something hard, they keep putting in, spikes and carabiners to protect themselves. Now we did that with the cysteine, right? But now that we know how to do that, just like in mountain climbing, people will do it, without rope. And so we don't need the cysteine. Now we can go for the aspartate. Now we can go for the valine. And we even figured out whole new ways to climb. And so that's kind of also going on. So RAS is being sort of the sort of template to put a lot of new drug discovery things that have happened in the last 10 years to work. So it's just a fantastic time. There's something we know we can do, and we have lots of new tools, and they're all getting thrown at it. I think right now that's why the curve is going so steep of our advances.
The resolution to get better, like I said, we went from the 110 to the 8K or whatever the technology, so we keep going. I'm sure those the crystalline structures, what is the device that you use to see that with? Yeah, probably that wasn't from the 1960s, was it?
That was right there. That's when the Nobel Prize for crystallography, those discoveries were made. But then, yeah, then they improved as well. Yeah. There's about 10 places in the United States where you can solve those structures. So, but they're big machines. Yeah.
Okay, all right, somebody is off of mute, so if everybody can just make sure they're on mute, doing background
Me in
noise.
there?
Uh-oh, let me see.
Okay, I'm here.
Okay, so hello, okay, we're gonna just keep rolling. We're just gonna keep rolling with it here. Let's see. Is it fair to say those with rare KRAS mutations like 61 will not benefit from the progress? I think we kind of like handled that.
We will benefit. Yes, those are another set we're going after for sure.
Yeah, yeah. And aren't some of those 61s are coming up in acquired resistance treatments?
Yes, I believe that's true too. Yeah.
Yeah, yeah. I just saw some data on that. I think I saw Margaret Waugh talking about it at ESMO over the weekend. See, which one is G12A? There are 3 A's. Okay, I'm gonna let you handle that alphabet soup.
Yeah, G12A is a known mutation, but 3A, I'm not sure, Lori, what you're
I think what I'm talking about is there's, and I have a little cheat sheet that I leave on my desk here, is like the F stands for phenylalanine. I'm brutalized.
Oh, I see. That's alanine.
Yeah.
There's 3
so the
A
different-
amino acids, alanine, arginine. Yeah.
Yes.
G12A, the A stands for alanine.
Okay.
Thank you, Terry. Wasn't going.
No, see, that's okay. That's why, like I said, it's just my job to not know stuff. Okay, and then any risk of binding of a cysteine on a different protein other than KRAS and non-specific binding? That is a good question.
Fantastic question. And that was part of the optimization effort of the drug to make sure that it could be dosed low enough to get onto KRAS, so the concentration wasn't high, so it wouldn't go do a lot of different reactions, and it would only go to KRAS. So there's that is a great question, and we can and have in the current drugs avoid promiscuous cysteine reactions.
Okay, so what I heard you say, okay, and seriously, correct me whenever I'm wrong. Okay, what I heard you say is that we want to make sure that it's hitting the KRAS tumor, not the healthy wild type KRAS that's wandering around doing the things it's supposed to be doing, and it's not getting promiscuous. Not a word I've ever heard referred to about my molecules before. Is that right?
That's right.
Okay.
I mean, we can make sure that the molecule does not hit wild type KRAS, but only glycine 12 to cysteine KRAS. But I think the question was asked about other proteins besides KRAS that have a cysteine. And that is an important drug design feature that was designed into the G12C drugs to avoid those other targets. Yeah.
I'm going to put the question, I'm going to read the question because it's next to my list, but I don't want to force you to answer. In your opinion, should KRAS drugs be used as first line against lung cancer and other types? It seems like that's more.
Yeah, I really couldn't tell you.
That's an oncologist question. Yeah,
thanks.
That needs to be depending on the trials and stuff. But it's a great question. And I anticipate that they'll be doing studies at some point in the future. So we'll be looking forward to those. Okay, Annie says, what are you seeing on the horizon for acquired resistance to KRAS G12C inhibitors? Dr. Corcoran mentioned the Revolution Med treatment.
Yeah, exactly. Ryan was the first one to publish a patient that had resistance emerge. And in that paper, he showed that the next generation drug coming from Revolution Medicines treated some of the mutations that emerged. So it was a drug that could be sort of leveraged in the next stage. Yeah.
Okay. So
great question. You guys know a lot about this.
We're really, I mean, we don't understand a lot, but we understand like.
Oh, you understand a lot. You understand a lot. I can tell from these questions. You understand.
Well, thank you. We have a smart group of people. Not only are we a good looking group, but we're smart. Okay, and so I really is a treat having you here. Let's see, how is sotorasib working with wild type KRAS and mutant?
Yeah, so sotorasib does not work on the wild type, but only on the mutant. The wild type does not have the cysteine that needs to be there for sotorasib to work. So because the cysteine is gone, sotorasib doesn't work.
Okay. Next up, I'm just going to keep going down. Thank you for your life-saving research. You probably get all sorts of thank you notes, but if you don't, then we will make sure that you do. Okay, could you comment on RMC 6236? That's the Revolution Med trial drug, the KRAS multi, and how the chemistry strategy differs from specific targeted proteins like the G12C example.
Great question. So the strategy of RMC 6236 is almost a completely new kind of drug. Now, what I mean by that is I've been telling you that you've got the key and it fits into the lock of KRAS. What the RMC molecule does is it goes into the cell and it first binds to a different protein in the cell. And when that protein is holding the key, which is the drug, it finds RAS and blocks it with, it recruits a whole different protein to RAS. So it's what we call a molecular glue because it's the glue between two proteins. And the reason for that, that was important to try to do is that when you have two proteins, you have a lot more surface to bind to rather than just a key, which is just a tiny little foothold. So it's like if you were rock climbing and instead of having 2 arms and 2 legs, the RMC drug is like 6 arms and 6 legs. It's just a lot more chance to hold on. And that was a very hard chemical problem because I kind of told you how hard it is to design one drug to hit one protein. They discovered a drug that binds simultaneously to 2 proteins and bring them together. So it took a lot more, not more work, but it's kind of why Justin's so excited about it. Because if you can get that thing to work, then yeah, I would rather climb a wall with 6 arms and 6 legs. I would go faster than anybody else.
Very, yeah, I think that's a really good metaphor. Okay. All right. When there's not a drug to target a KRAS mutation, does KRAS typically respond better to immunotherapy or chemotherapy? Again, we're talking molecularly here. We're not talking like because you're not an oncologist, you're not a doctor, so you're not doing it. So what do you see?
Yeah, I don't know, and I bet it's very specific to different tumor types and what the other mutations are. So your point there is I don't find good comparisons, and that is because it's very hard to compare apples to apples. That's a hard thing.
Okay. Let's see. That's so helpful. Thank you. We should all pressure companies pursue that avenue. I'm not quite sure what that was about, but
maybe the IV, maybe.
Oh, maybe so. Maybe. Oh, you're just staying on top of this. I should just let you just do this. I'm just navigating. Where are we with the KRAS G12D colon cancer trials? I think we keep coming back to that.
I think they'll be at the end of this year. My guess is they probably will come out in a slew of them. I bet there'll be a bunch of them opening, I hope this year, but certainly by next year from what I hear at meetings.
It seems like it follows school year. Okay.
Really?
Wow. Now again, this is not based on some scientific analysis and data. I've really only been paying attention for 2 years, but it feels like it's about that. I mean, if it doesn't happen by the end of May, it's not happening until September. It's kind of how it feels. I don't know. Maybe I'm totally wrong. I don't know. Maybe somebody.
No, you're probably right. Yeah.
Probably somebody could write a white paper on that. Okay, I have tears of gratitude. I want you to know, I get emotional when I read some of this stuff and I hear about this. And then I get to meet people that are doing this. This is amazing. And Lori agreed. Another Lori said, I'm a cancer survivor and a protein crystallographer. That's a thing. I understand how hard it was for Dr. Shokat and his colleagues to accomplish this. If I can be a resource to anyone in this group, Lori, let's talk. I'm not sure what
Lori.
Crystallographer is, but I'm figuring it's kind of the person who keeps track of all the crystal structures, kind of like a stenographer would for a
Oh no, it's like Lori can probably give a different analogy or a better one, but it's like the best chef you ever had that was also a nuclear scientist and a mathematician, because you need to make the crystal and then you need to go to the source of the atoms to bombard the crystal. And then you need to be a mathematician to then convert that into the three-dimensional picture. So,
well, we're
Lori,
really wild, Lori. How come you need to reach out to us? Let's have a conversation. Okay. And so yes, you can be a resource, Lori. Thank you for raising your hand. Okay. Sandy says, are you familiar with NCT 051984, an Amgen trial with Lumakras infected for colorectal cancer? That's not really kind of like, we can kind of like help you find something,
I don't know.
Sandy. Yeah, that's kind of like outside of this conversation, I think. RMC 6236 is multi-RAS drug by RevMed. I believe it can be used for different, I think we kind of hit on that earlier.
That's exactly right. That's the kind of target for that.
Yeah.
Yes, exactly.
Okay, if the molecules of adagrasib and sotorasib are ever so slightly different, does that mean the molecules of different drugs can have slightly differences which affect their efficacy? It's a really pointed question.
It's a great question. And, that's, they're very close, but there are differences, and people won't, it's really hard to figure out where they come from, what the differences are manifest in. We know some things, that some resistance mutations to adagrasib at the position 95 that occur will become stay sensitive to sotorasib. So there are sort of single residues we know that will emerge in response and cause resistance to adagrasib, but then they will still be sensitive to sotorasib. So those are some concrete differences, but in terms of tissue and dosing, it's sometimes hard to know. Again, comparing apples to apples is hard.
Okay. All right. Oops, I just lost. Okay. So Annie says, we talked about binding to the pocket and HER2 molecule tips into Herceptin. Are there other ways of attaching to a cancer mutation? So like metaphors like Velcro or sewing or knitting together, or I kind of like what you're saying with the false hands, and how I envisioned it was kind of like putting a molly in a wall to, because you don't actually have like a stud you're hitting, right?
Right.
And so that's kind of like you put the thing in the wall to kind of like spread out, and then we can kind of get at it. And you like that one. Okay, good.
I do.
Okay.
This question is really great, because there's another way to attach to a cancer mutation. And it kind of takes this, what I told you before, one step further, which is like, some things are reversible. They bind and they unbind. And then the G12C drugs, they bind and they stay stuck. They stay stuck for the life of the protein. So until that protein gets chewed up by the cell, that drug is on there and the protein's off. There's some drugs that bind and slowly can come off, and they don't come off of the intended target at all, but they come off of other targets in the cell that they accidentally get stuck to. So there's just so many cool things in drugs that we're learning about and designing that, so it kind of gets to that knitting question. I don't know what the right metaphor is for that one, but we call those, we have reversible drugs, which I told you about. We have irreversible covalent drugs. And then there's this group in the middle that are called covalent reversible. So they make the bond, but they fall off sometimes.
Okay. I'm picturing like, it's almost like getting double-sided sticky tape on you, and it's like you're just trying to get it off. It's like,
Right.
you gotta keep going somewhere, right? Okay.
Yeah.
All right. Let's see. Clinical trials open in Canadian patients is kind of like outside of scope here. How much work has been done on G13? Let's talk about G13. I think we kind of hit like in general, like the G12s and but is there something that's really so complicated about like being on a different allele, being on the 13 instead of a 12?
Yeah. We thought that the G13 would be the next target we go after. We went after G12C, and then we worked on G13C. And boy, when we tried everything, we never got anywhere. So just that one position away, it kind of oriented the cysteine from towards the pocket to away from the pocket. And that little difference made us not be able to solve that problem. But when you start to apply these other approaches, like the 6-handed, 6-foot, approach, then you can get after G13 mutants. So there are, I know, G13 programs. So those should be coming.
Okay, and you're saying this is a silly question. This is not a silly question. Is a KRAS mutation genetic? The testing wasn't around when my grandparents had lung cancer, and I wonder if there's a genetic component. Can you address any of that?
This is a great question. There's two kinds of genetics that we often don't say what we're talking about. There is a genetic predisposition, which is a sort of your genome might have some different, you have different genome from your parents and your kids and things, and little differences might give rise to a little bit higher chance of a cancer. That's not the kind that KRAS is. We have this is the other kind of genetic, which is it's a mutation that occurs after you're born, and it occurs only in certain cells. So the genetic predispositions are in all of your cells, but the ones that occur later only occur in certain cells that then grow to become the tumor. So the genetic component could have been partly in the family, but it wouldn't be on RAS. And then you might have gotten RAS separately.
Okay, so a tendency, like my family, cancer runs in my family. If somebody dies from heart disease, it's a shock, but cancer runs in my family, not necessarily one kind, and it may not all be KRAS, and it could be any other heart. And so it just, there's some other proclivity there.
That's right. We think that those are often mutations that occur in a different kind of gene called the tumor suppressor gene. And so sometimes the tumor suppressor pathways that are always protecting us from cancer, maybe in some families, one of those protective mechanisms is a little less active. So that means that we're a little bit closer to getting a cancer if we get a new mutation. That's the hard part.
Okay, yeah, and I saw that there's been some stuff that's been coming out on ATM genes and that sort of stuff as well.
Yeah, we're working on that. We're working on reactivating P53 so that you could get a lot of the cell to help you treat the cancer. So that's exactly.
Yeah, I think that's kind of one of those things that's kind of developing.
Exactly.
Is right. I mean, that's the fun thing about doing these is like we could do this like in a month from now. Things could be different. I mean, and that's what's cool. That's what's cool. I mean, it's frustrating in some ways, but it's what's cool too. Okay, are these compounds too large, or why are they unsuitable for inhalation or sublingual to avoid microbiome variability? That's a good question. Why can't we just take them by mouth or just up our nose?
Yeah, no, sublingual is a very interesting route. I mean, that's how nitroglycerin is given for patients with angina, and it acts really fast. Not all drugs enter that route, or the ways you can formulate the drug into a pill don't lend them that way. Inhalation is also a possibility, but it takes different chemical properties. And overall, a lot of tumors might be moving from the lung, so you want the drug to be everywhere too. But those are great questions.
Okay, all right. What's the difference between the work you're doing and going after Panras? That's another good question.
Great question. We decided to not go after Panras because we wanted to be tumor specific. And so all of our strategies in my lab are making covalent bonds just to the mutation that drives the cancer. But I'm thrilled that people are making Panras inhibitors because that could turn out better. It's just kind of, we have a limited number of people in the lab, and just kind of the way we don't like to, in the lab, we can't compete with a company that has 100 or 200 or 1,000 people. So we try to take approaches that are a little bit, unusual or off the beaten track, and hope that we, find somewhere new. Yeah.
How big is your lab?
14 people now.
Okay, that's a good number of people.
Oh, it's a lot.
It's not like having 100 though, I tell you.
No, exactly.
Yeah, I hear you. I hear you. So you know what I mean when I say that, like, we're little, we're just going to be mighty, right?
Yeah.
Okay. Let's see, differences in responding to inhibitors between yeah, I think that's kind of, that's an oncology question, oncologist question. Fourth quarter is deadline for a lot of companies for their stockholders. I think that was in relation to when clinical trials are coming out. Yeah, I mean, we do have to play the shareholders. I mean, that's just the, you know, reality of it is if it's not
A
financial.
lot of the companies are private, but you're right, the public ones, there may be considerations.
Okay, I had a message from somebody who says, thank you for what you're doing. What you're doing is directly saving this person's life. And she's getting scans, and it'll be 2 years since her initial diagnosis. I mean, honestly, Dr. Shokat, you really do not realize, like, I mean, the fireworks we would have, okay? And I'm in Carolina. We know how to have fireworks down here. Okay, we don't do fireworks like they do out west. We do fireworks down here. Okay.
All right, I got to see that.
No, seriously. I lived in Arizona for a long time. They don't do fireworks. There's one person with a sparkler runs around a circle. That's it. Out here, I mean, they like almost blow the whole city up. It's crazy. But no, seriously, it's a big deal, and we really do think having you here is really making a difference. And so we really appreciate it. So don't stop. Okay. That's not me just, that's not like, okay, now you're done. No, keep going. That's me applauding and saying, keep going. Okay. Let's see, is there any data taking KRAS G12C inhibitors before HIPEC? I think that's more of an oncology question, isn't it?
Yeah, it is. I wouldn't.
Yeah, that's kind of like more of a clinical trial question. It's a perfectly good question, but this is a little bit different. See, it's a patient, is there a downside to covalent irreversible bonding? Does it prevent you from using? That's a really good question. How does it impact a patient? I mean, you're seeing it. How does it impact a patient?
Yeah, you know, right now, I mean, 10 years ago, people didn't want to make covalent drugs. They wouldn't develop a program if it had a covalent drug because they thought the downside was potential for toxicity because it might attach to other proteins and cause an effect. But we've learned a lot from the drugs. One of them is in B-cell tumors for the kinase BTK. It's called ibrutinib. And that one is quite well tolerated. And so we used that as our guiding principles behind the design of these G12C drugs. So, you know, once one drug works, then you can sort of leverage that understanding. But you're right, at the beginning, you might think an irreversible drug has more chance of toxicity.
Okay, all right. And I'm looking at my clock and we're like, right, I mean, I said it's still 7:30 my time. And so I don't want to like be disrespectful of your time because
I'm fine.
Your dance card is like really, really full. And so I really do appreciate you being here. And I just, does anybody have any questions that they really just like want to shout out? Because otherwise I'll just like, I'll just start asking questions on the fly here. Right. And then we never know where that's going to go. Okay.
Somebody asked about, they found a tumor with BRCA mutation.
With BRCA2 and KRAS G12C. Is the G12C just mutation destructive and harmful than the BRCA2?
Yeah, it's a really great question. You know, that's BRCA is partly a you know, tumor suppressor. It corrects kind of DNA damage. So it's really hard to say, is it more important, you know, for the tumor, the accelerator or taking off the brake? It kind of is probably different. It's probably, each of them are probably important at different stages of the cancer. So at the end of the tumor, now that it's developing, it's hard to know which one was more important. They were probably just equally important. Okay. But in terms of which one we could drug and how we, you know, we could, that's a really good question.
Okay. All right. Do you think the open relationship of the RAS initiative is helping to accelerate progress and sharing of research? Well, my opinion, not that anybody asked my opinion is yes. How about you? What do you think?
Oh yeah, because they created, they kind of could solve problems that nobody really could solve before, like making the authentic full RAS protein with all of its modifications and being able to give it to people to study. We often in the lab chop up the protein and study just a little part of it. And sometimes that's just because it's easier. But at the RAS initiative, they can say, no, we want the full thing and we want to make that and we want to send it to people. So it really accelerated solving hard problems and providing reagents. And the meeting that they held every year is a great place to see the cross fertilization of ideas. Yeah.
I'm planning on attending this thing because I mean it's not that far from me. And
it's great.
Yeah, it doesn't mean I'm going to understand anything any more than some of this other stuff, but we're doing what we can do. See, whoa, my screen's jumping around. Why do you think the map pathway has so many fail-safe mechanisms?
Wow.
Oh, that's real speculative.
Great question.
Wow, let's go. Yeah, get into it.
I mean, I think it's because, cells rely on that to grow, and so when there's so much part of that pathway is a homeostat, is like your thermostat at home, and it wants to be at a certain level. And once you try to push it too hard, then it will compensate back. And if you inhibit it, then it will try to come back to the middle again. Why it is biologically, boy, it's hard to answer evolution questions. You really just don't know what it was selected on, but it's a really deep question.
Yeah, and it is. It is. Okay, success in lung cancer for G12C versus colorectal cancer. Chemically, can you give us any thoughts on that?
No, I don't think chemically it's an answer. I think it's a biological difference between the cells that arise from colon cells versus arising from a lung cell. And there's a lot of differences in the tumor suppressor differences between the two. So they're just sort of different branches of a tree, and one might be easier to break off and one might be closer to the trunk. It's really hard to know.
Is there a way that you can study it? Like, I mean, it seems like there would be like some version of like a microbiome that you can do like chemically to be able to analyze it and see the differences. Is that?
Yeah, we're doing that now. A lot of people are, but you can sort of put a tracer in and watch the colon cancer cells respond to the G12C inhibitor and the lung cancer ones and see what the differences are. So we've got sort of molecular flight recorders, if you will, on cells, and we're watching them. So
and as a patient, that gives me a little bit more confidence too, that they're checking it out, in a test tube before they're just like dumping it into my human body too. Okay, let's see. Do you think beacon trial with multiple MAP kinase pathway inhibition is relevant for KRAS and NRAS? Annie's going to keep.
Don't know that specific trial. Yeah, sorry, I don't know that specific trial to give you a good answer.
All right. Let's see. Lastly, thank you for giving so much of yourself for progress. We are grateful. And so probably we should be thanking your wife and your wife and kids as well.
Oh, yeah.
I know how family life works, and I have no doubt that they gave as well.
Oh, yeah. Yeah. Especially when things don't work in the lab, it's always nice to come home and think about something else before you go back in the next day.
Yeah. No, seriously. I mean, that's kind of how life is, right? Let's see, at Harvard Mass Gen, they're looking at pulsing. So going through between chemo and targeted treatments. Do you have any insights into that?
That's an idea that emerges every few years, and it has a lot of appeal. And there's a lot of preclinical data that makes you think that the pulsatile will be really differential. But as I've seen it, when people have tried that in the clinic, then it doesn't really pan out. But it's such a good idea that I think we have to keep trying. So I don't know about that trial specifically, but I keep an eye on that area as well. I think it has it's got to work somewhere. So I think it's great.
Well, so chemically, then it stands to reason that it would make a difference potentially as to the order of treatments and the way things happen and way it would unfold. I mean, if it makes a difference when I'm making cookies, it's certainly going to make a difference when it comes to killing the cancer.
Yeah, and I think partly the vision for the next 10 years is to use the circulating DNA from tumors earlier in patient trials. So when we give a drug, we look what's happening and we follow that, and we can pivot before we waste time on a drug that didn't work and get onto a drug that will work. And when it's working, to keep driving it and, you know, try to get beyond get more molecular pictures of what's going on as the drug is in the patient.
Okay, now, do you know the difference between RNA and DNA? Do you see any, talk me through the significant differences that you would see like with some of these therapies?
Oh, you mean how
yeah.
the DNA would respond?
Yes.
Yeah, well, when we kill a tumor cell, it breaks apart and its DNA is then, you know, handled in the bloodstream. And so we can sequence DNA for long times because it's a stable molecule. When we look at responses of a treatment in tissue culture, we often use the RNA levels, but that's an unstable molecule. So we often look at circulating tumor DNA in a patient, but we look at the RNA levels in cell culture. So those are kind of differences, yeah.
Okay, all right, I don't know where, my screen just went goofy on me. Okay. Okay, let's see what else. So thank you for sharing. Thank you for, we really are grateful for you.
Well, thank you. You guys taught me so much. I mean, I know, Terry, last time we talked, you had such great questions, but now I see that the group you've put together, they have, so you guys are asking questions that I hear at, you know, from Nobel Prize winners on advisory committees. So you guys are fantastic.
Well, you know what, but we are 100% of the patient community that we care about, okay?
Yeah, that's so
to a doctor, we're one of thousands, right? And so we really want to be able to understand it, and we really, you know, want to be able to, you know, educate ourselves, right? And we learn from each other. And so I really appreciate you taking your time and being able to speak with us. And I do look forward. I hope we can grab a cup of coffee or something.
Yeah, let's do that.
And you know, and all that. And I think it is just going to be a fantastic. And it's like, let's see, what on earth is this? Oh, thank you, Terry. Wait a minute. When you're not studying KRAS in chemistry, what are some of the hobbies you like to do to relieve stress? That is not from me. That is my husband, my other laptop is what that says. And so
I like to bike ride. I like to bike ride. I commute by bike, and then also weekends go on bike rides. Yeah.
Okay. Well, from all the analogies, I would think it was mountain climbing.
I wish I was not strong enough.
Well, you need to get the right, what was it, the right picks in there, and then
That's
you
right.
need to get that device. And I'm kind of envisioning, what was it, like from one of the Batman movies or something, where he had like or I don't know, one of those sort of movies where he had like a superhero thing on. But
Yeah.
Thank you so much for joining us tonight, and thank you guys all for being here. I mean, when we bring folks in, and I'm just overwhelmed and wowed by the amount of energy and knowledge that we're getting from you to the community. And it's just spectacular. And we really better together, because you really are out there doing what you got to do. So
yeah, well, it's all for you guys, all for the patients. I think every day the lab members come in, that's, you know, really what keeps you going through all of the hard things, because we've all had friends and family with cancer. So I'm just glad that we've been able to make some contribution.
Well, thank you, and keep doing what you're doing, and just kicking KRAS, right?
Yes.
Okay, for sure. Okay, guys. Thank you
Take
all.
care.
All right. Take care.
See you in New Orleans.
Okay, sounds good. Okay, bye. Bye, everybody.
Thank you, doctor. Thank you, Terry.